Hello everyone, and welcome to our lecture today. Today we will be chatting about microbial growth, binary fission, and we will be looking in depth at the growth curve. Just a bit of a heads up, this will be a shorter lecture, just given the nature of things, nature of the content. So yeah, you guys are in for a bit of a shorter one, hopefully, unless I go down too many tangents. So first off, I think it's pertinent to start off at the basics and looking at what reproduction is and reproduction and growth, specifically as it pertains to microbes. This might seem like a silly question, but just keep in mind that when we talk about microbial growth, we can refer to two things. Microbial growth can refer to cell growth, which is simply an increase in the size and the weight of individual cells of the individual organisms then. Or it can refer to a population increase. This would represent an increase in the numbers of bacteria. So generally, when you will read any texts, be they journal articles or textbooks, when we talk about microbial growth, we tend to refer mostly to population increases or increases in numbers. That's the more common use of the term. Then, so then how do these organisms reproduce? Well, the primary process is through binary fission. This is simply an asexual process where one cell divides to form two daughter cells. So one parent cell becomes two daughter cells that are genetically identical to each other. So in this instance, as I mentioned, this is a form of asexual reproduction, meaning that there are no gametes involved in this form of reproduction. Then there are two other reproductive processes I really want you guys to be aware of. That's budding and fragmentation. So budding is also a form of asexual reproduction in which a new individual will develop from some generative anatomical point of the parent organism. And then fragmentation, which is also known as splitting, is also a form of asexual reproduction. You might be catching a trend there that the reproduction here is always asexual, where each fragment will develop into a mature clone genetically and morphologically identical to its parent. So essentially, you have one organism that just splits up into many. The organism may develop specific organs or zones to shed that may be easily broken off. This refers more to more of your microbial animals, obviously. And then another process that you'll see I marked it here with a bunch of question marks, because this is maybe one that you wouldn't think of normally. If you think of endospore germination, remember endospores are those resistant states that these microbes can enter, almost like a form of hibernation, if you want to look at it that way, where, if conditions change and they become no longer conducive for this microbe to thrive, the microbe can enter the state to become just resilient, to just essentially hang in there until its situation or its environment becomes conducive to its growth again. So as I said, endospores, under these suitable conditions, they will then germinate, and the resultant vegetative cells will grow and reproduce then again by binary fission. So you'll go from this endospore stage back to binary fission, where it'll just continue on and grow and grow and grow and grow. So interestingly, the life cycle and survival of bacilli are dependent on their gram-positive cell wall structure and their ability to form endospores. So think specifically of them when we are referring to this process. So I'm going to walk you through basically what binary fusion is. Again, it's just as an overview, it's an asexual reproduction where the parent organism is separated into two new bodies. In the process of binary fission, an organism duplicates its genetic material, so its DNA, and then divides it into two parts, which is termed cytokinesis, with each new organism receiving one new copy of the DNA. So they will be clones of each other. They will be exactly the same genetically. So this process will start first with the elongation of the DNA. So you can see here the clump of DNA is being elongated, and you can almost start seeing it. You can see where it will start to separate. And then the cell wall and the plasma membrane will make these you can almost think of them as little kips. They will start dividing in to pinch off, essentially. And here you can see that process, what it looks like from a real organism's point of view. And then following this, that cell will start, the cross wall will finish its development, will finish its growth, and it will surround the DNA, and then the cells will separate. So there you have your one parent organism that has now divided into two daughter cells. And again, these two daughter cells will be genetically identical. This is also why in bacteria or in microbiology, we speak about a colony forming unit, because all of the members of that colony will be genetically, if you isolated it well, will be genetically identical, especially if they originated from one single cell, because they're just making clones of themselves. Okay, and just to give you a better idea of what this process looks like, because often when we think reproduction, we think mammals or plants, which is a slow process. But if you look at bacterial or microbial growth, it results in, this binary fission process results in an exponential growth. So you will have one parent cell that immediately becomes two daughter cells that will then also divide, making four cells, who will also become two cells each. And like you can see, this exponential growth means that if you look at the numbers, the concept of doubling, that you have you can have a very rapid increase in these organisms. To give you guys an example from organism that I'm pretty familiar with, given the amount of work I've done with it. If you look at E. coli, its generation time can be as little as 20 minutes. We'll be talking a bit more about generation time later on, but just keep that number in mind. So every 20 minutes, if I can just jump back here, every 20 minutes, under very ideal conditions, you can have two daughter cells from your original one and your bacteria. So every bacteria in your original sample will then have formed two daughter cells, so your population would have doubled in 20 minutes. This is just a representation of that. So remember here, this power that you see over here, so it's 2 to the power of 5. Remember, anything to the power of 0 becomes 1. And then 2 to the power of 1 will be just a 2. 2 to the power of 2 is, of course, 2 times 2, so it'll be 4. 2 to the power of 3 will then obviously just be 2 times 2 times 2, and so on and so forth. I'm not going to run through all of that with you, but what I want to point out here, why I went through that, is this exponent number here is the number of generations. So we refer to this as the 0 generation. This is the parent organism, that's just a single one. And the first generation will then result in 2 organisms. The second generation will then result in 4 organisms, and so on and so forth. So that number that you're using there as your in your calculation of number of cells, that number there is your number of generations. So why do we use exponents to express the growth? Well, here is a very good graph showing you exactly why we do this. So here you can see on your x-axis for both the y1 and y2 axis represents generations. Here on this y-axis we have number of cells. Keep in mind that this is not a logarithmic scale, so this represents this thick line over here. And then on this side, we have a log 10 number of cells. So this is an exponential scale. And this results in this straight line, which is way easier to visualize then. But keep in mind that these intervals are exponents. So this represents 10, that represents 100. Everyone represents a 10-fold increase over the preceding one, where here you have normal intervals, which results in this very hard to interpret, if I were to ask you to do some calculations between the 0 and 5th generations using this graph that is not a non-exponential, it would be very difficult to actually visualize and see what's happening, because it looks like no growth is happening, and that is definitely not the case. So that gets us obviously down to the question, what exactly is this generation time? What is the actual definition of it? So generation time is the average time interval between the birth of an individual and the birth of its offspring. So essentially, how long before this organism can make, in this case, daughter cells, but can make babies? How long does that time period take? So generation time can depend on, obviously it can vary between different organisms. Remember, I mentioned the E. coli example earlier, between like 20 and 40 minutes that it has that generation time. But other microbes will have different generation times. Some can be much quicker, some can be slower. It'll also vary depending on the growth conditions. So but here's a concept that you will definitely obviously find in your textbooks, but I think it's important for me to just mention it here, that remember that under optimal growth conditions, your generation time will be minimized. It sounds counterintuitive, but it makes a lot of sense if you remember that generation time is the amount of time between when an organism is born and when it can have its first, daughter cells spawning from it. So the quicker that time period is, the quicker your population will be growing. So the shorter that time period is, the quicker your population will increase. So under optimal growth conditions, you will have a very short generation time. So the organism will be able to replicate very, very, very quickly. If conditions start becoming more adverse to them, they won't grow as quickly, obviously. So now we're going to look at how to calculate generation time. This obviously is relevant, and this may come up in your exams, so I expect you guys to know this. In our calculations, these will be our variables. So generation time will be represented by G. You can see G is there. So G is equal to T over N. So what is this T and N? T is simply the time interval, and N is your number of generations. So that makes sense. So how much time is spent making these generations? You must, this may be a bit above this and beyond the scope, but if I give you an equation like this, typically I will allow you to assume that generation time remains consistent. This might not be the case, but because it's an introductory course, I feel it's fine for you just to know what these variables represent and how to calculate generation time. So this N, the number of generations, can also be broken down by this log that you see over here, where this small b is representative of the number of bacteria at the end of the time interval, so when you stopped measuring, and then the large B will be the number of bacteria at the 0 time. What this means is just when you start your bacterial count. So this is your initial bacteria numbers versus bacteria numbers at the end. So initial, end. So if you look at some examples here, if you're running through this equation and how to use it, you can then rearrange the equation based on the variables you're given. So if you're given a equation such as this, you can rearrange your equation, calculate, and arrive at your generation time. And this is how we actually change the equation, and that's why we use the 3.3 times log. So I'm not going to go into too much detail for this. I don't want to go above and beyond. Have a look at it. I probably won't ask you to explain the math. As long as you are able to apply the equation, I feel that's good enough. So that gets us to applying the equation. So here this is a nice example. Something like this might appear in your exams, where I will provide you with a large B. I will expect you to know what this large B represents, and this represents your initial bacterial count, or your initial bacterial concentration or population number. And small b is your number of organisms at the end of your study. So in this case, large B, so you had 100 cells at noon, so at 12, and then you had 100,000 cells at 5 p.m. And then I will ask you calculate the generation time and calculate the number of generations. Or I might just ask you to calculate the generation time and expect you to know that for that you would need to first calculate the number of generations. So how do we do this? First, you'll have to figure out the time. So obviously between noon and 5 p.m. there is 5 hours or 300 minutes. Keep in mind that you typically want to keep your time in minutes, because these bacteria have such quick generation times. You don't want to be dealing with 0.4 something of an hour or whatever the case might be. It's much better to be working in minutes, just as a tip. You can always convert it at the end of your calculation, as long as your numbers are correct. I don't care how you do it, but just as a general tip, it is generally easier to work in minutes because these organisms tend to have very quick generation times. So how do we go about this then? So first, n is equal to 3.3 times the log of b over b. Remember, this equation is from that previous slide. Don't expect you to know how to derive it. That's why I didn't go into detail for it. Then we will be subbing in our variables. So first small b, so our concentration of bacteria at the end of the study was 100,000. We'll divide that by the initial, which is 100. And then calculating this thing out, we'll get to 9.9, which you can then approximate and round up to 10 generations. So more or less in this time period, to result in this type of increase, you needed 10 generations to divide, essentially. Then you will take that number, the 10 generations you just calculated, and you will divide it by the number of the time. Remember, because your generation time is simply the time interval divided by your number of generations. Thus, you will have 300 minutes divided by 10 generations. So you'll have roughly 30 minutes per generation. Again, I won't give you any more complex examples than this. I will keep I'll keep the conditions similar, unless there's a stated question where I ask you calculate it under these conditions and then calculate under these conditions, but those will be two different calculations. I will supply with that, so don't worry about that too much. Now we're going to move on to something that's really important that you guys have to understand. So we're going to be spending quite a bit of time on this, and that is this bacterial growth curve. So bacterial growth follows a sequential series of growth phases. There are actually 4 of them. That is the lag phase, which you see down here, the log or exponential growth phase, which you see over here. This is the second phase. Then you have a stationary phase before you have a death phase. Keep in mind that for ease of for you guys to understand and just to visualize this better, this is all on a log number. So this is all exponential. And then something else that I want you guys to be familiar with, because we have discussed staining before, bacterial staining. So I want you guys to know this. During this death phase, what do you guys think will happen if you look at these organisms under a microscope? What will happen? How would you distinguish between what's living and what's dead? Remember, we discussed viability staining. So, moving on to our next slide. This is a classic example of viability staining. So just as a reminder here, viability stainings differentiate between live and dead bacterial cells in a sample. The fluorescent stains used here base the differentiation on whether or not the cytoplasmic membrane of the cell is intact. So if the cytoplasm is still being contained in the cell, that is what the staining is based on. So cells that are still appearing green on this stain are considered live cells, and cells that are red are dead cells. But if you were to throw this under a normal bright field microscope, the cells would look like cells. So you have a very hard time to distinguishing between the different bacteria, which ones are alive, which ones are dead. That's why we do viability staining, to realize how much of the bacterial population is actually still alive at the time of investigation. So let's move on to the lag phase. I will be discussing all 4 of these stages, and we will be starting with the lag phase. So what happens during the lag phase? So during the lag phase, and so there's very little or almost no cell division, and that's why it's called the lag phase. Essentially, in the lag phase, these organisms are like rearing to go. They're like getting ready to grow. They're like adjusting to their conditions. They've now recognized, for example, that the conditions are now, optimal. Conditions are now good to grow in, and they use this lag period to get ready to start growing. So what do? This means that they will increase their cell sizes. They might adjust their metabolisms, and it's a time of adjustment for them. So essentially, these organisms used to be living in some sort of unfavorable environment. They kind of struggle to survive. So now during this lag phase, they also have to start sensing that they are actually now in better environment, be that because of temperature, pH, nutrient conditions, whatever the case might be. They now start to realize, hang on, we can actually, go ham here in a second. So they start gearing up for growth, and this can involve also, with metabolism changes and structural changes. They can, for instance, start making ribosomes. And this lag phase can take anything, from a few hours to a few days, depending, how long if it's a culture, how long it's been in storage, or how long the microbes have been in a unfavorable condition. It takes them essentially a while. You can always think of it as the population waking up, or getting ready. And that's all that happens down here. That's why you see there's no real increase in the population numbers yet down here, because these organisms are still sensing what's going on around me. Can I start, do I have what is needed? Do I have food? Do I have the correct temperature? Do I have the correct pH? So I'll start growing, and all of that will happen here. That's why you see no growth. And then they'll start growing, and then what happens next? Now you have your population boom. Now they grow and grow and grow and grow and grow. So essentially here, your division is at a constant or a maximal rate. And this is technically, when you calculate generation time of an organism, this is when you want to do that. You want to calculate it during this interval, during this phase, because it's this is when the organisms are actually really trying to divide. They're actively trying to increase their population numbers. So, and keep in mind again that this is a logarithmic increase. Remember how the 1 cell became 2, became 4, became, that's how these organisms are going to start growing now. And then also, you obviously, as you might imagine, cellular reproduction is most active during this period, and generation time reaches a constant minimum. Remember, you want your generation time. If conditions are optimal, your generation time will start becoming smaller. So this is when it'll be constantly at its minimum value. So this population will just grow and grow and grow and grow and grow. And that's what happens here. That's why you have this explosion of numbers. Keep in mind, again, this is a logarithmic scale. So this is a really, really, really large increase in numbers. And when you are calculating the generation time, or the maximal growth rate, you want to calculate it when this line is almost linear. And another thing that I want you guys to keep in mind, I'll be doing a little throwback at the end of this curves, but this would also be a very, very good time to do your gram staining, because all of your cells are typically still going to be healthy and intact. They wouldn't have started dying yet. They're still like just growing. There's still enough nutrients, there's still enough space. They haven't really produced any waste yet. So this is a really good time to do your gram staining, because the cells will generally, the individual cells will still be quite healthy. Now moving on to the stationary phase. So in the stationary phase, the name here might be a little misleading. Just keep in mind that stationary here just means it's like in an equilibrium, where your reproduction rate is equal to your death rate. It doesn't mean that reproduction stops. Under no circumstances is that what that means. It just means that the rate at which the cells now are now increasing is equivalent to the rate at which they are dying. So essentially, yeah, the population is now starting to stabilize. It's starting to reach this equilibrium, the stationary phase. You will also obviously now start thinking as a microbe, well, there will now start becoming some nutrient depletion. In your environment, the nutrients are starting to get used up by all your competing siblings. And there's also some toxic waste being built up, or it could be start being built up. I mean, it's these microbes, same as any other organism, they produce waste, and this waste could be harmful to other members of their family, extended family, if you want to call it that way, or if you want to look at it that way. This toxic waste can also be toxic to themselves, obviously, and then obviously their cloned brothers and sisters. And then, so what do you think as a microbe would be a pertinent thing to start doing when you start seeing this turn in the conditions? If you start thinking, oh wow, I don't have any more nutrients, there's some waste, there's some toxins being built up. If you can make endospores, be a pretty good time to start doing that right about now. So this is what you see exactly here. This flattening out of the line. I want to stress that this does not mean that cell growth or population growth has individual organism growth has stopped, or division has stopped. Binary fission hasn't stopped. It just means that the death rate is equivalent to the increase. And then the organisms that are capable of doing so, the bacilli that we spoke about earlier, will now start forming endospores. Then we move on to the death phase. Now you have your population crash. This is also an exponential process, or an exponential rate, because you have to keep in mind that these organisms cannot manage their resources. They are not cognitive in that sense. If the resource is there, they're going to use it until it's done, and then they are just going to all die off. That's what's going to happen here. They can't save the resources or budget their population or plan in any way, shape, or form. So they're going to keep increasing until they use everything and have taken up all the nutrients. They've produced way too much waste. They've taken up all the space, because keep in mind, space can also be a limiting resource. A bit of a curveball there. But that could also be the case that there's just no more space. They can't grow anywhere anymore. And then they will be this population will be decimated, because there's just no more resources. And the population just simply cannot be sustained. So this phase will then continue until the population is diminished to a tiny fraction of the numbers in the previous stationary phase. It can even result in a complete decimation and eradication of the population. They can all die out. And that's what happens over here. So your death rate now starts to greatly exceed your growth rate, and the population just absolutely plummets. And this can continue on until the population is completely dead, or they can just sort of stabilize or remain at a, especially the ones that formed endospores, will remain at a concentration way lower than they were in the previous stationary phase. So remember when I said gram staining is a good idea to do your gram staining here? Why do you think that is? Why do you think gram staining now might be problematic? It's very simple, because there's a lot of organisms that are dying, and that the fact that they're dying, they might be bursting open, and they might not be able to contain their cytoplasm. And why would that be a problem? Well, if their cell wall has failed or is compromised, the peptidoglycan will not be able to hold the crystal violet anymore. So you will be mis-IDing your organisms because they're dead. You're not staining live organisms. So yeah, that was a quick little look at, not a quick little, but that was the look at the 4 phases of growth in bacteria. Something that I want you to keep in mind when reviewing this lecture: keep in mind the structural or physiological changes that might occur within individual cells during the growth phase, during the lag phase, during the death phase. What would the individual organisms look like? Because that is important. It's important for terms of like your gram reaction, as we just discussed. When would the endospores form? So if you want to do endospore staining, when would be a good time to do that? And other testing, including like the susceptibility to antibiotics. Something that I do want to keep talking to you guys here about a bit. Remember, as I said, these organisms cannot plan. They have no sense of self-awareness or self-consciousness. We don't think they do, at least. It hasn't been shown to be the case. But what that means is, so these organisms, just to review, will keep using resources if the resource is available. And that means that if you continuously add new media and you remove old growth and toxins and waste, these organisms can essentially grow indefinitely. So, case in point, there is an experiment called the E. coli long-term evolution experiment, or LTEE. So this is an ongoing study on experimental evolution led by Richard Lenski that has been tracking genetic changes in 12 identical, well, at least initially identical populations of asexual E. coli since 24 February 1988. So they essentially had 12 populations of bacteria that were genetically identical, and they've continued to grow those 12 populations, obviously in isolation from each other, to look for changes in their genetics, changes in to how these organisms evolve. And just a few freaky numbers here. So the populations, the 12 populations, reached 73,500 generations in early 2020 before they were frozen and stored because of the COVID-19 pandemic. So there's been more than almost like 74,000 generations of this bacteria grown since February 1988. That's pretty insane. So then the next question becomes, what are some of the ways that we can monitor microbial growth? How do we know bacteria, the growth is occurring? How do we know how much is happening? So what I want you to I hope you guys can remember back when we discussed the work of Louis Pasteur and his contributions. Remember that when bacteria or fungi or archaea grow in broth culture, they make the solution turbid. It becomes hazy, fuzzy. So here you can see how that progresses from clear to sort of murky to completely turbid. So we can use this to determine how this organism or this population is growing and increasing in number. How do we do this? We use a spectrophotometer, and this device measures the absorbance at a 600 nanometer, not meter, nanometer wavelength. So essentially what this instrument does, you can see it represented over here. You have a light source that is then allowed to shine through your flask. Here's a blank one, and you will see, just think of these lines as representing the intensity of the light. So here, with the blank sample, you have a lot of light intensity reaching your light sensitive detector on your spectrophotometer. Now, if there's a lot of organisms in here, what's going to happen? So essentially, the number of bacteria will decrease the amount of light that's transmitted from the light to the spectrophotometer. See, now there's only one line, so the intensity of the light will decrease with greater number of organisms, resulting in less light being transmitted. So there's an inverse proportionality there. And we can measure that and then use that to determine growth. And just as a final reminder, just remember, the less light that is transmitted, or the less light that reaches your photometer, the more bacteria is in the sample. It's an inverse proportionality. So here's an example of how that was used. This absorbance at 600 nanometers, good Lord, 600 nanometers was measured, and this is the amount of time in hours, and this is how they plot the growth then. Here you can see it was also compared, comparison of the growth curves of Chlorella vulgaris, which was grown in 3 different media. So this was in a complete medium, media deficient in nitrogen, and then media deficient in sulfur, represented by these 3 lines, as you can see over there. So, and there you can just see an image of what this algae actually looks like, because this is an algae. So what I want you to also take away from this image is this is a very good example of why you might want to be using a chemically defined medium, remember from our last lecture. And this is because you can increase and you can control the amount of nutrient concentrations and see how it impacts the growth rate. You can alter the nutrients that the microbe has and then see what that does to the growth curve, to see what nutrients in what quantities is needed to sustain whatever microbe you are investigating. So something here I want you guys to know, this is that relationship that we were speaking about, the OD600, which is an abbreviation for the optical density of the sample at 600 nanometers, and the colony forming unit. So this is you can think of this as the number of cells, number of organisms. There's that proportionality I was speaking about, and this is used to then estimate how quickly the organism is growing. You can see there's a very nice proportionality there, where you have the more organisms leading to more an increase in your OD600. So you have that nice, almost, I mean, it's approximated almost as a 1-to-1 relationship, but it isn't. It's just you have this nice straight line with a very, very highly significant R squared value. So that's why we can use this method to investigate population growth. So here's a nice example of how we would do that. You can do this experimentally established relationship between colony forming units and absorbance. Remember this graph over here. And then you can solve for CFU. So if this is your original equation, and this is derived from your line, the gradient of your line, that's how you can view this. This is where we derive this equation from. And then you can rearrange this to solve for CFUs, which is colony forming units, think number of organisms. And then you can, if we are supplied with numbers, then that is what you can use to calculate your number of CFUs. Alternatively, we can also count them, of course. So this is just a silly little example, but we can actually count microbes, and it's done quite often. But you might also think, well, that creates a problem because these organisms grow so quickly. There can be thousands of millions of, there can be a lot of them. So how do we do this? The answer is, of course, dilution. We will dilute our original sample into a manageable number, and then we will calculate back to our original concentration. So, for example, if we do serial dilutions and plate counts, you will have an original inoculum. So this represents our original sample, and you will take 1 milliliter of that and add it to 9 milliliters of broth. So this gives you a relationship of 1 to 10, a dilution factor of 1 to 10. And then you will repeat that. So then you will take 1 milliliter of your now diluted sample and add it to 9 milliliters of broth again. This will give you a factor, a dilution of 1 to 100, and so on and so forth, 1 to 1,000, 1 to 10,000, 1 to 100,000. So then you will be plating out your inoculums and incubating them, following which you will count the number of colonies on them. So there's an important thing here. So when you count these organisms on the plate, you can't just take any one of them. You have to use the plate that has a number between 30 and 300. Anything in excess of 300 is too much, and anything less than 30 is too few. So if you have 3 like this, you can't use that. You have to refer to the previous one. This can also be done using the pour plate and the spread plate. Remember, we discussed that in our previous lecture a little bit, and I asked you guys to read up on that, so I'm not going to spend too much time on that. But this is how we will prepare the plates that we will be using for the plate counts. So I want you guys to really review this process in your textbooks. It's not really that difficult. Remember, the primary difference is when you're using the molten agar versus when you are spreading the inoculum over the surface. There's also videos kindly provided by Dr. Shen when he taught the course, and he demonstrates how to do these techniques. They have been posted, and you will be discussing them and doing them in the lab section. That's why I'm not spending too much time on them right now. But just please be aware that this is how we will be preparing the plates that you will then be using in your dilutions. Now, moving on to today's review questions. I was asked by a few students to perhaps provide some alternate examples, maybe of some different ways that I could ask some of the questions, maybe even provide a few long form, or solutions to longer questions. And that's why I included some of the stuff I did today. So a question like this could be, present in the midterms or the final exam, where I'm asking you, apart from endospore formation and binary fission, define and explain 2 microbial reproductive processes. And this will then be for 5 marks. You can oftentimes, as a tip, before I'll give you guys some time to answer, use the mark allocation to determine how much, more or less how much you want to write down, because for this you're going to have to define something and then provide an explanation. So that'll be 1 mark each, and then you'll have to do it for 2 processes. That'll be 4 marks. So there's something else that I need. So providing the answer to this question then, the 2 processes I was looking for is budding and fragmentation. And then providing me with all this detail, I'm not going to read all of this, but this would be representative of a long answer that you would get full marks for. So that would be 5 marks. Moving on, I'm going to ask you another one here. Calculate the CFU concentrations in the sample of river water for 5 marks. So here's your sample of river water. You've done these dilutions, and those are your plate counts resulting from the dilutions. So I want you to tell me what the original concentration was, or the original colony forming unit concentration in the river water was. I'm going to give you a couple of minutes here before moving on to the answer. Not minutes, sorry, seconds. Keep mispronouncing things today. Units of measurement, so difficult. Okay, I'm going to be moving on. So the correct answer here, there's a few things that I want you to keep in mind. Remember, I said you need the concentration between 30 and 300. So the TNTC is too numerous to count. That's just you had too many. You had way more than 300. 140, that falls slam bang in the middle. But if you use this 18, you're also incorrect because that's too few. You have to have 30 or more. So you would simply just calculate this back to its original concentration by calculating this dilution. So you would have a 10-fold, 100-fold, 1,000-fold, 10,000-fold, 100,000-fold dilution. So you would take this concentration, multiply it by the number of dilutions you've done, and then that would provide you with your answer of 14 million colony forming units. Remember, per millimeter, really important here. So there's quite a few things that give you marks here. The first is knowing which number to use. The second is correctly calculating the number of dilutions that have been done. And then there's also, I want you to give it as a concentration because I asked for a concentration. So I expect the answer to be in the form of concentration or CFU per milliliter. So using the correct unit, really important here. You couldn't just write down a number. You had to use the correct unit. So number, unit, what number to use, factor of dilution, all of that together gives you 5 marks. As a general tip, I was also asked by a student last week if I will be providing with study guides. The answer to that question is no, I will not. My reasoning for that is all your exams will be open book. Your class tests will also be open book. So I expect you guys to prepare beforehand, to make notes, review the study questions in your textbooks. They're there for a reason. They're good practice. Make sure you go over them. When you, the same can be said exactly for the labs. The lab exams, you have ample time to prepare them. I expect you guys, I've posted all the lab sections, or most of them online now. We still need, I think it's lab section 8 and 10. Those will be up within the next couple of weeks. Don't worry about that. You'll have enough time. But I expect you guys to review that material before you attend your labs so that when you are in the labs, you can ask Amy and Kieran questions. If I am there, it's difficult for me to attend because of my other obligations. But if I'm there, you're welcome to ask me, of course, what you want to know too. I'll happily answer that. So I will not be providing study guides. I expect you guys to prepare. You have open book exams. If this course was taught under normal conditions, the exams would not be open book, so you already have a massive advantage there. And I think I can't stress enough how important it is for you guys to prepare for your tests and exams because they will be set up in such a way that you will be pressed for time. You will not be able to refer to go digging into your textbook for every answer. You have to know what is where in your textbook. I don't expect you to memorize the entire thing, of course, but if you have prepared beforehand, if you've done the exercises, you've made notes, you've made little, use this little sticky notes, and you can quickly find answers in your textbook. That's what I want. I want you to know where to find answers. I want you to know the work. It's not about parroting it back to me, but I want you to understand, and for you guys to understand the work, you have to prepare, you have to study, even if it's open book. I can't stress this enough. Okay, guys. Lecture aside and little advice and tips aside, I will see you guys in the next one. So thank you so much.